Hey students, practical is very important in biology. It guides and prepares the student towards understanding the subject of biology. Practical is very essential in the field and also in the laboratory. Now, if you have to go through practical in biology, there are certain tips that you are supposed to know. First, you start with observation. For anything you are given in the lab, you require your senses to observe first. You need to observe very carefully. Look through. And by going through, by looking, that is the observation or examination, there are certain things that you may require the use of a microscope or hand lens to magnify your view, where the specimen provided are in minute or small size. Because you are supposed to see clearly, and after seeing at the end, you are supposed to draw exactly what you see. So the first thing is for you to observe. You use your eyes to see clearly whatever you are provided with. Now, whatever you are provided with is what we call the specimen. The specimen will require you to specify the type of the view you are supposed to give. The view could be dorsal, that is the upper view of the specimen. It could be ventral, the lower view of the specimen. It could be lateral, the side view of the specimen. It could also be posterior, that is the end view of the specimen. It could also be anterior, the front view of the specimen. You may also be required to give the transverse section or the cross section of the specimen. So you have to look and specify the type of view you are supposed to give. After doing that, then you are supposed to give the title of whatever specimen. Now, whatever you do, I said there is need for diagram. You have to draw. Then your drawing must bear a heading. That gives you the title. The heading is stating what it is and its scale, its size. It gives you the title. The size and the magnification is also very important in biology class. Each drawing should bear a magnification. And this magnification is simply the size of the diagram and the actual size of whatever specimen you are drawing. It helps the examiner knows the real size of whatever you are drawing. All diagrams in your practical biology must not be less than 8 centimeters. However, you must keep strict to the specification given by the examiner. If you are asked to give the size to be 10 to 12 centimeters, you must keep to that instruction. Your diagram must not be less than that size. That gives you the magnification or the size. And then we have the level and the leveling. Very, very important. In level and leveling, there are two basic things. We have clarity of line and neatness of level. Clarity of lines gives you the real lines that are used in your diagrams. They are supposed to be drawn by sharp-pointed pencils. They should be single, they should be thin, and they should be continuous and non-thickening. In no way should you thicken your lines as you draw your diagrams. Then the neatness of level. After your diagram is drawn, then you level. While leveling, you are supposed to use a ruler to draw your lines to make them neat. That is where the neatness of level comes into action. Now, it is very essential that you use a ruler to draw the lines that you are going to name. The lines should touch the parts to be named. As you draw the line, it must touch that particular part you are naming when you come to level. Lines must not cross each other as you draw them. Each line should be on its own. They should not cross each other. There should be no use of broken lines. Use a straight line with your ruler. And then levels must be written clearly and horizontally. They must be written clearly and horizontally. Do not label both sides, all sides of your diagram. You must keep to the horizontal side. This will help you as we go along with our subsequent practicals. For now, we are going to take the food test, and on the board we have the table for what we are going to test. The first column is the test for starch. We are supposed to collect any starch material. For this particular class, we are going to use bread. Bread is a common staple food in our homes. On daily basis or frequently, we come across bread. We take bread at any time of the day as part of our meal. Now, we are going to use the bread to test for the presence of starch. Then, I have the bread here. I'll use the test tube. I'll crumble some parts of it. My colleague earlier has said that we use solution in most of our practicals. So this particular one is also going to be in solution. We are going to add water so that we can make it in a liquid form. We have the water bottle. We add water into it. And then the instruction is that we add few drops of dilute iodine solution. We have the iodine solution here. We use the dropper to get some few drops. This is the dilute form already. Then we drop. And on dropping some few drops, can you notice the color? You can see now we have a blue-black color. So now if we have this, then it is showing that starch is present in this bread, having the color. So in our observation, we write a blue-black color is observed. And then we conclude, we infer that starch is present. Now the second test is for simple sugar. We are supposed to put a small quantity of glucose solution in a test tube. We have the test tube here and we have the glucose here, laboratory glucose. We take some few samples and we want it in solution. We have the spatula to collect. We're supposed to have it in solution. We add some few drops of water. And then the second instruction is that we add 2% of Benedict solution. This is Benedict solution. We still require another dropper to collect. Thank you. We drop in, and let's observe. Now we are supposed to heat it for a few minutes. It requires heat for us to get what we want. We have our source of heat here. We have a cylinder. We have a Bunsen burner here. Then we heat. You are supposed to heat or to boil for a few minutes. Can you see it? As we heat, a brick red or orange precipitate is formed. Can you see? We have an orange precipitate. You can see the mixture looking a bit thick, not an ordinary solution. That gives you the precipitate. So this concludes that glucose is present. If it were a different color, then there is no glucose. Are we okay with this? This is the glucose test. A brick red or orange precipitate. We have the orange precipitate. If it gives us brick red, it's still in order, it's still okay. This is orange. Now we have to go to the next solution. Test for proteins. We'll go for the Biuret test. We're supposed to take a small quantity of fresh milk solution. We have it already in the beaker. We take small quantity of it. We take another we have to take a small quantity of fresh milk solution. Then we add 1 ml of sodium hydroxide. This is sodium hydroxide. We add it up. We have the sodium hydroxide, and then we will now have a drop of copper 2 tetraoxosulfate 6 solution. This is copper 2 solution. We'll still add to it. Now let's see. You have to keep observing. We shake, and what color are we seeing? The mixture is changing to purple. This is purple. So when the mixture turns into purple, it shows that protein is present in the milk. Can you all see the purple color? So this signifies that proteins is present in fresh milk. We use fresh milk solution. We added 1 ml of sodium hydroxide, and we added a drop of copper 2 tetraoxosulfate 6 solution. And we have seen the change in the color. It was initially milk. It's turned into purple after the addition of these reagents. And this infers that protein is present in fresh milk. Is that okay? Good. Now we test for oil. This is a filter paper. You can use any available white paper. We have oil here. We drop some of it on the paper. We just have some few drops of oil on a white paper. This is a filter paper, very common in the lab. And then the translucent test allows us to view this paper through any available source of light. We look at it through the window, and you can see the spotted parts looking transparent. We look at it through any available supply of light. We look at it through the window, and we can see the center where we have the oil spot looking transparent. Now this transparency is what we refer to as the translucence. This is the translucent test, and it shows that oil is present. Earlier, my colleague has introduced you to those three major food parts: carbohydrates, proteins, fats, and oils. We have gone through certain food substances in our houses, common ones, which shows that these materials are present. We have run a simple test using starch material, bread, to see the presence of starch. We have tested for simple sugar in glucose. We have used the laboratory glucose. At home, we can equally use the cane sugar juice. Cane sugar. You can collect the juice and you can run this simple test and you can have this particular result. So if you don't have the laboratory glucose, you can use simple cane sugar, which is very abundant in our society. We have seen the test for protein where we use fresh milk solution plus sodium hydroxide and copper 2 tetraoxosulfate 4. We have tested and we have seen the presence of these food materials in this food. So let's go back to the table. The first column, test for starch. Collect starch material. It could be bread, but for this particular class we have used the bread. It could be yam, it could be any other starchy food, but for this purpose we have used bread. We diluted it, we put some water in it to have a solution. We added few drops of dilute iodine solution, and we have seen the reaction. The color changed into blue-black. That is what we observe. And that observation refers that starch is present in our bread. We have put a small quantity of glucose solution. We used the laboratory glucose. We diluted it, we dissolved it to have a solution, and then we added the Benedict solution, and we boiled, we heated it using our source of heat, the Bunsen burner, and then we had an orange precipitate being observed. The orange and the brick red gives the presence of glucose. We have tested for proteins. We took a small quantity of fresh milk, we added sodium hydroxide solution and copper 2 tetraoxo 6 solution. We have observed the mixture turning into purple, and then that purple color tells us that protein is present in the milk. We have equally done the paper test, translucent tests. We tested for fats and oils. We have some drops of oil. This is vegetable oil. We had the drops and we looked through the available source of light, and the transparent part shows that oil is present. So it is very important that we know these things, we carry them carefully and gradually, and we have our best result. If we keep to the instruction given, and the examiner will definitely give you the table. Test for a particular thing gives you the procedure, and now it is left for you to follow the procedure strictly and then observe what will come out of it. Your observation gives you your inference. That's okay for now. Until I come your way next time, remain blessed.